My computer says 9 o'clock. Let's go ahead and get started. Number of people have been asking me about clickers. Most of you have your clickers registered and everything is happy and hunky-dory and all that good stuff. Yes, today they will start actually counting towards a final score at the end of the term. Keep an eye on D2L. I'll just be uploading all those scores hopefully every day. If you see absolutely nothing there, then send me an email with your Odin username and the clicker number. Those are the things that I'm going to need. A couple of people have different names in the registration for the class and registrations for the clickers. Maybe you've changed your name, I don't know. So I just want to make sure that those are all correct. So if you have questions about clicker registration, this is a great opportunity to introduce yourself to your neighbor, because they will probably have registered their clicker and it seems to be working. So with that, we'll move through today. We'll finish up talking about proteins in a general sense, then start talking about our favorite model here, DNA, and the structure thereof, or I should say the model of the structure thereof, and how Watson and Crick were wrong in a lot of cases. And then we'll see how far we get with that. We'll talk more about the chromosomes next time in class. So as an outline from what we talked about last time, proteins just in general, amino acids, as we'll see also with the nucleic acids, have a molecular biology polarity to you. They've got an amino terminus and a carboxy terminus, giving you a front and a back. Those then hook together with peptide bonds. One amino group hooks up together with one carboxylic acid group. Those then make an extended chain, but they're always going to have one amino terminus and one carboxy terminus. Through hydrogen bonding interactions between those backbone amino acids, and I say backbone atoms on your amino acid molecules, those are going to be the ones that are part of the peptide bonds. Those hydrogen bonds are going to form the secondary structures. They're going to be alpha helices, beta pleated sheets. And we'll see, particularly the case of beta pleated sheets, a little bit later on today. Those secondary structures, in and of themselves, are usually not stable, just that particular piece. It's only when they come together in terms of a tertiary structure, your three-dimensional structure, then you get stability. And it's that process now of the side chains interacting with each other, which is going to give you those kinds of structures. Structures are going to lead to functions, yes. We talked a little bit about the difference between domains and motifs. Again, this is something that I want to emphasize because it seems to be unclear, probably unclear the way I talk about it, as well as also in the literature. A domain is a part of a protein which has a specific structure, which is going to have a specific function. That has to have a structure in and of itself. Just the, if you were to cut out the whole rest of the protein, usually with proteolysis, that particular domain is going to have a stable structure and a stable function. Motif, on the other hand, is something you see in primary structure or primary sequence. You're just looking at the stretch of amino acids from N to C terminus. If every 7 amino acids you see a large hydrophobic, something like leucine, then you can guess, and this is a guess from that sequence motif, that that's likely to form a leucine zipper. Every 7 amino acids in an alpha helix, you're going to be sticking that same amino acid side chain on the outside. So that's your motif. We'll talk about other motifs later on, but the motif is just from the sequence, where you can guess what it's going to do. Any more questions on that? Because again, this seems to be a complicated concept for some people, although maybe I'm getting better at explaining it. I had a question on D2L about multimers versus quaternary structure versus multimeric proteins. Basically, all that we're going to talk about here, and it turns out that the vast majority of proteins are this way, they're more than one polypeptide chain. So they've got an N terminus and a C terminus. Each of those individual polypeptides then comes together to form an active structure. It can be a homomultimer, i.e. two of the same protein. When I say the same protein of the same sequence, or they can be heteromultimers where they're different proteins with different sequences that come together to have the overall activity that we're looking for. And the big one that we looked at there was the DNA-dependent RNA polymerase, which, depending on what organism you look at, has 12 to 14 individual polypeptides that come together. And I'll often talk about these as subunits. So each of those subunits you could also consider as one of the mers that's going to come together in the multimer. Protein families, we talked about this before. This is, their process of either speciation or gene duplication gives you protein families. The important thing about that really has to do with similar sequence. How similar that sequence should it be if you want to think it's going to have the same structure? We're all happy with this? Yes, no? Generally, about what? We'll see. And then we talk a little bit about modeling protein sequences, et cetera. That's still very, very hard. So going from a sequence to structure is still practically impossible to do. So questions on what we talked about last time? Yes, no? When he says, are there any questions, what does he mean? He's about to ask you a question. So clickers, again, for the marbles, as it were. Let's get this on. Our next question is proteins that have at the following percentage of amino acids. Obviously, I didn't edit this. Proteins have the following percentage of identical amino acid sequences. Most probably have similar structures if they have 15, 25, 35, 45, or 55%. And I would say the minimum number there. So have we started yet? No, it's still not starting yet. Okay, I need to get rid of this before. Now we're going. Yay. Yes, I'm talking about the minimum. A lot more people have clickers today. We're at 130, 140, 141, 2. Okay, 142. There are 175 people, I think, registered for the class. So few of you don't seem to have clickers, or somebody's got a whole bunch of clickers that they're voting for. So what do we think? We're split about half and half between 25 and 35%. So what do people remember, if at all, about what I've mentioned in class? 30% is usually the cutoff. So 30% would be 25 is lower, 35 is higher. This is also a really good example of the kinds of questions that I love to put on my exams because I'm lazy, and having answers that are numerical are really easy to come up with the wrong answers. So you'll notice this on a number of my exams. So the correct answer here is 35 because it's above 30. Okay. So did want to talk, this is actually exactly the same slide that I used last time, but I went back and listened to my notes, which my wife said, that sounds like your voice coming out of the computer. So I think I didn't do a terribly good job of explaining this, so I wanted to come back and revisit it again. The basic idea here is, we've talked about our secondary structures, alpha helices, beta-pleated sheets, etc., tertiary structures, wonderful stuff. It turns out that, again, about a quarter of proteins have generally no fixed structure unless they're associated with something else inside the cell. That could be another protein, like we have here. So the squiggle here is supposed to mean it doesn't have a fixed structure. This green one means that you do have a fixed structure. So it could be just these two proteins interacting with each other. This doesn't have a standard structure by itself, just random, what people call random coil, i.e. not a fixed structure. It's flopping around a lot. Here, if it interacts with another protein, then it has a defined structure. Often, it'll be some kind of change, and we'll talk about these covalent changes, phosphorylation, a little bit later on. That will also give you now a fixed structure, and I would say this should be green now. You can also have parts of proteins, so this would be like a domain of a particular protein, that under some conditions will have a fixed structure like down here. Under other conditions, it won't. And then there are even a few proteins in the cell that never have a really defined structure. And so that's this example of pores, sort of a diffusion barrier, et cetera. And until, I don't know, maybe about 10 years ago, most people thought this wasn't really true. You didn't have these unstructured proteins, just didn't have the right conditions for them. But it does turn out that there are quite a few generally non-structured proteins that you'll find in the cell at any given time. Another thing that people thought for, again, a long time, if you had one amino acid sequence that gave you one structure. This is also not true, and probably best known of these cases are the, this thing is still deciding not to work on me properly, are the amyloid proteins. Probably most of you have heard about amyloids, which is a particular structure of protein that forms in neurodegenerative diseases. So Alzheimer's, Parkinson's, and a number of others, particularly best known for BSE, bovine spongiform encephalopathy, mad cow disease, where you have one protein which can then change the structure of other proteins, which is really bizarre. And again, nobody thought this was true until Stan Prusner actually got the Nobel Prize for showing that it really was the case. But what happens here is you have a protein, see here on the left, with what people often would call a low complexity domain. What do we mean by low complexity? By low complexity, we mean sequences which repeat a lot more often than you would usually expect. You don't use all 20 amino acids in this particular case. And it turns out that a lot of these low complexity domains, regions of proteins, can then make beta sheets with each other, and often two beta strands. Then these two beta strands can come together to make these much larger structures, and it turns out that sometimes the presence of these two beta strands can help other proteins to form these two beta strands, and this is the problem with BSE and probably also true for Alzheimer's, Parkinson's, et cetera. So first, it was all the nasty stuff. We don't want amyloids at all. This is another way of talking about it. Amyloids are these multi-beta strands which form. But then more recently, people have also found that these kinds of multiple beta strand structures are also very important for concentrating things inside the nucleus. There are these things called granules. Granules is just a little dot that cell biologists used to see on the inside of cells and go, oh, it's a dot. We'll call it a granule. And only much later they figured out that these were often places where different subcellular components were being concentrated. RNAs, we'll talk a lot more about RNAs later on. They get concentrated in these granules, and a lot of that has to do with this amyloid formation. Low complexity proteins forming these two strands of beta sheet, which then in multimers can form these very large aggregates as being amyloids. So these would be the good amyloids, the bad ones, of course, the ones that are leading to these neurodegenerative diseases. A couple of people last time asked me about, well, how can you have something which is only 30% identical at the amino acid sequence and still have a more or less identical structure, and then, of course, more or less identical function? Most of that is because for the vast majority of proteins, only a very small proportion of them is actually involved in the activity of that particular protein. Classic example of this is enzymes, but if we go back and look here at these amyloids, what's important here about these amyloids? What's important is this structure down here. that they're interacting with. It's not all the rest of the protein, it's just this smaller piece. And we'll see this is true for the vast majority of proteins, that you have, for instance, all this gray part on this protein, which is, still trying to get our technology to work here. The gray part of the protein, this sequence actually really doesn't matter that much as long as it brings these few amino acid side chains right here into exactly the right place. So when you talk about sequence conservation, i.e. the amino acids which have to be the same in the whole family of proteins, which are all going to have the same function, the rest of the proteins, the rest of the amino acids is really not that important. However, when you do have those particular amino acids here in the yellow circles, the red circles, and the blue circles, those are absolutely critical for the enzymatic activity. They're going to be the binding and interaction part of that particular protein. So the idea here is not so much, oh, this is cyclic AMP, which is interacting with glutamic acid, etc. It's that the vast majority of the protein is just there to get a few amino acid side chains into exactly the right place. And that's how you can have these proteins that have very different amino acid sequences, but still have basically identical functions with them. So in most cases, again, it'll be one small part of the protein that has this very high conservation. But there are some proteins that have two different parts of the protein that have very well conserved sequences. Now, why would that be? Well, that's because you have communication across the protein. And so a lot of examples we'll talk about through the rest of the course have this sort of action at a distance. One part of a protein talks to another part of the protein. And when I say talk here, of course, that's totally anthropomorphizing, but it means that what happens on one part of the protein is really important for what's happening in the other part of the protein. And we'll see particularly this is important for multimerization again later on as we move through the course. But the idea here, and this is an example for enzymatic activity, is you have a protein, in this case a dimer, that has an active site, which is going to be here, glucose, which is this blue hexamer. In this open case, it's not active. In the presence of another molecule that binds to some other part of the protein, it leads to activity. And because of this allosteric, meaning change in one part of the protein that's separate from the first part that you're interacting with, that allows us to have an activator molecule that will activate this particular enzyme. Exactly the opposite thing can happen here as well. We've got an allosteric regulator, i.e. it's binding to the protein away from where the active site is. Again, the active site are those specific amino acids that you need for the enzymatic activity that we just looked at. Far away, something that interacts there that changes the activity. And if it's changing the activity, changing the function, what else is it doing? It's changing the structure. So we'll look at some more examples of this as we move along. This is a very general kind of case here, very important for enzymes, but as we'll see, also important for signaling molecules that we'll be talking about in this class, and particularly important in cell biology as well. So let's talk about some of those examples here. Basically, what we're trying to do is we're trying to change the function of a particular protein. How are we going to change function? We're going to change structure. So a number of different ways of doing this. These are the most common ways. The most common is phosphorylation, which is taking a phosphate and putting it onto one of the amino acid side chains. This is a covalent change that happens. Putting on a phosphate, really onto any macromolecule, is the reaction called a kinase. So it's putting on a phosphate. Taking that off is a phosphatase. We'll talk a lot about ACEs as we move through the rest of the class. ACE means enzyme, and 90% of the time we're talking about an ACE, it's going to be important for hydrolysis, which is going to be doing what? Adding water and splitting things. So ACEs, usually, again, with some exceptions, are going to be breaking things apart. So phosphatase, what does the phosphatase do? It takes phosphates off. So kinases put phosphates on, phosphatases take phosphates off. Why is phosphate such a wonderful thing to use for this? Phosphate's reactive. What else about phosphate? PO4. What are those O's? Have negative charge to them. And so we've talked last couple of lectures about interactions between side chains, interactions between different molecules. Charge-charge interactions are often very important for that. And so if you change the charge by putting a phosphate group onto something, that can really change a lot the other proteins that are going to be interacting with that, or for that matter, nucleic acids. So phosphorylation is very common here for these kinds of changes. Again, it's a change that happens through a covalent modification, again through addition or removal of a phosphate. This happens, again, through the activity of kinases and phosphatases. You add a phosphate 99.9% of the time from ATP. That then changes the structure, gives you a signal. A protein phosphatase, a separate protein, will take that off, and you have now a change in structure and a change in function. Probably the other major form of this change in structure, change in function, has to do with what are so-called G proteins. G proteins, also known as GTP binding proteins or GTPases. These bind, not surprisingly, to guanosine triphosphate. The binding of guanosine triphosphate will change the structure of this protein, giving it some kind of other interaction, some kind of other signal. What's very different about the G proteins versus the proteins that are getting phosphorylated is these actually turn themselves off. So the hydrolysis of GTP here, unlike the phosphate that gets taken off by the phosphatase, this happens through this G protein. This is also why people call these GTPases. ACE enzyme, what does it do? It breaks up GTP. In most cases, that's not the only thing which is having GTPase take place. Very often you have a GTPase activating protein that speeds up the enzymatic activity of this G protein. But you don't actually have to have this. On the other hand, what you do have to have is a guanosine nucleotide exchange factor, a GEF. Because once hydrolysis has happened, now you're bound to GDP, guanosine diphosphate. To get that diphosphate off and triphosphate on, you do need another protein in this case. So difference here, kinase and phosphatase, you have to have two enzymes here to cause this interaction. With the GTPase, you just need one, which is exchanging this nucleotide. This phosphorylation, here I say the phosphate gets removed spontaneously by the GTPase. So let's look at a couple of examples here. This is just kinase phosphatase. Kinase puts phosphates on. Phosphatase takes phosphates off. Here's just an example. Serine is a very common. We'll talk more about these: serine, threonine, and tyrosine. What's the similarity between all of those amino acid side chains? All got OHs, which is very good then for putting phosphates onto. You can phosphorylate other amino acid side chains, but they're much rarer. So here, serine, threonine, tyrosine, then get phosphorylated, and the protein, that phosphate group gets removed, the activity of a phosphatase. This change, the addition of a phosphate or the removal of a phosphate, can either activate your protein, which is what's shown here at the top, or it can be inactivating the protein. And that just has to do with what that protein is going to be interacting with. And so when you say turned on, turned off, that's going to be just what this particular protein is interacting with. So in this case, phosphorylated is on. In this case, phosphorylated is going to be off. Again, just both ways that this can happen here. Let's look at a case of a GTPase. This is actually probably the best studied of any of the GTPases or GTP binding proteins. This is RAS. This is a protein which is mutated in something like 50% of solid tumors, which gives you an idea how important it is for general cellular development. We can talk about where that RAS mutation is a little bit later on. But this is the normal RAS protein bound to GTP. When it's bound to GTP, this is the structure of RAS. Once you have hydrolysis that takes place here, this piece in red moves and basically just moves a little bit closer here and changes whatever interactions are going on here. So GTP binding is going to change the structure. GTP hydrolysis is going to change the structure as well. So here we have a schematic of what's going on here. These are both the same. This could be just a circle drawn the same way. When bound to GTP, this is active. You have GTP hydrolysis. Again, this can happen in the absence of any other protein. Then you have the inactive form. GDP is released. Usually this is going to be stimulated by that extra protein, the guanosine nucleotide exchange factor. Bring in GTP, you'll have an active protein. So this is actually different. The fact that you don't need an extra protein here to get the change between active and non-active means that GTPases serve a lot more as timers. So once you start this activity we have up here, GTP hydrolysis is going to happen. And so you'll be on for a while and then turn off a little bit later. On the other hand, this kinase process, you have to have another protein. So once you flip the switch, turn it on or turn it off for that matter with adding the phosphate, that is not going to change until you have something else which comes along and puts on a phosphate, takes it off. So kinases, I'll be with you just a sec, Dinah, is flipping on a switch, flipping off a switch. That's what happens with protein kinases and phosphatases. GTPases is you flip on your switch, but eventually it's going to turn off. Yeah? So the question is, does the release of GDP happen in the absence of help with these guanosine nucleotide exchange factors? Yes, it does, and it's shown on here as being very slow. In almost all cellular reactions, that will be far too slow to actually have this be fundamentally useful inside the cell. So almost always you have a nucleotide exchange factor, which will help speed up that. But it can happen completely independently. Yeah? Mm-hmm. So I'm not quite sure I understand your question, but I'll sort of repeat it. Hopefully that'll help, the explanation process. So getting back to Diana's question, that this release of GDP and binding of GTP, that can happen in the absence of another protein, but cellularly, that would be far too slow to make this a useful kind of reaction. So you'll need that extra protein to help that exchange process. So getting off the GTP, getting off the GDP, excuse me, and putting on the GTP. But that can happen spontaneously. In the case of kinases and phosphatase, no way. It's not going to happen in the absence of any of those. So that's a big difference between the two. I have to get a different remote here for this, because this is causing me all kinds of problems here. OK, so this is just, again, sort of repeating the same thing which I just talked about. If we can get this to work. Technology. Have to love it, right? So just looking at another kind of GTPase, many cases GTPases here are multimeric GTPases. RAS is actually a little bit of an exception because it's just a single domain ATPase. Most of the ATPases that we'll see in molecular and cell biology are actually multi-domain GTPases. So the change in the structure in the red helix, which is up here, is not going to change interactions with separate proteins. It'll change interactions with separate domains in the same protein. And that is what you see here. Here we have a G protein, GTPase, multiple different domains. This domain up here, domain 1, at the top is very similar in structure to what we just saw with RAS, that single domain protein. Here in the GTP-bound form, this red helix, again it's the same red helix, is interacting with different domains of the same protein. As soon as you have hydrolysis here, this helix moves relative to everything else, and then you have changes in these other domains. And this is also one of these things you can talk about, so I talk about allosteric regulation. The allostery here is we have a change in one part of the protein, which then causes changes in other parts of the protein. This could also be true again for phosphorylation. If you've got phosphorylation that's happening here, clearly changing the charge where you have interaction between two different parts of your protein is going to change how those different protein parts are relative to each other. The last protein modification that I wanted to talk about before we move on to nucleic acids has to do with the ubiquitination system. Hopefully people heard about ubiquitin. Yes? What the heck is ubiquitin? Good. Okay. So ubiquitination is usually thought of as, I like to call it the eat me signal, so protein degradation process. Once a protein has a ubiquitin that's been added to it, it then gets degraded. That's what people thought it all used to be important for. Turns out ubiquitin is important for lots of different processes inside the cell. Also can be used a lot like phosphorylation as just a signal. It's a covalent change that happens to a protein that then, in the case of ubiquitin, it's a, remember correct, it's about an 80 amino acid extra protein which gets added to the protein that you already have. Clearly adding a whole new domain to your protein is going to change the function of that protein. So how does that happen? Turns out you need two different enzymes to do this and one sort of adapter molecule. So just want to step through this briefly. Those of you who know Dr. Singer, this is the main focus of the research in his lab. So the way that this works is we have three very creatively named proteins, E1, E2, and E3. E1 is called the ubiquitin activating enzyme. What it does is basically makes an activated intermediate. So we've talked about activated intermediates already. This activated intermediate is binding to ubiquitin, again, through the activity of ATP hydrolysis. Now this activated intermediate interacts with the E2 protein. This is the protein which is going to add the ubiquitin to your green protein, the target protein, which is going to get changed. This is basically just the transfer of the activated unit from E1 to E2. The reason that this happens appears to be that E2 can interact with E3. Now, E3 is not an enzyme. What E3 is a specificity factor. And so, what's specificity factor? Specificity is if you're going to modify a particular target protein, like this green one, you need to modify the correct one. And that's what these E3s are all about. And E3s, basically it seems that for every different protein that's going to get a ubiquitin modification on it, it's going to have a different E3. This is your specificity factor. So E1 and E2, what does E1 do? It puts ubiquitin onto E2. E2 is going to put ubiquitin onto another protein. All very non-specific. This could happen for any particular protein. In order to make sure that ubiquitin goes onto the correct protein, then you have to have the correct E3. And so this process, E1 goes to E2, goes to E3, ends up giving you a ubiquitin with this little red question mark, which is present on here. In many cases, there'll be lots of ubiquitins which are added here. And this then usually is going to lead to protein degradation. But it's only one of the things that can happen with this ubiquitin modification. And again, because Dr. Singer does a lot of these, and we've got nice examples in the textbook to talk about this, wanted to zoom into this E3 protein here. So this is an E3 protein down here at the bottom in these multiple different colors. The green part is the so-called culin protein. And again, culin is what Dr. Singer works on. It interacts with a number of different proteins, particularly this F-box protein and adapter protein. So what would you call this? E3 proteins are what? They're multimers, and they're heteromultimers. So we have the E2 protein, which is going to interact here. This is now the enzyme. It's got ubiquitin hung onto it. This then can transfer that ubiquitin to whatever is binding to this E3 protein. This particular part of the E3 protein, this is where you're getting your specificity from. And down here it's supposed to show you've got two different specificities, two different parts of this culin protein, which are going to allow you to bind to your target protein. These are just a couple of different examples here. So this is how ubiquitin signaling works. Again, we're sort of advancing in difficulty, or complexity, I should say, between phosphorylation and GTPases. These are relatively simple, few numbers of proteins. To the ubiquitin system, we are going to need multiple proteins, but basically to do a very similar job. Covalently change or change the structure of one protein in order to give it a different function. So because of all these different interactions, said, culins interacting with F-box proteins and all of these other things, in order to understand how all of these proteins are functioning inside a cell, it's often very useful to know what they're interacting with. Yeah?
On the last slide,
yeah.
So for each of those ubiquitins that are put onto your protein, is a new E2 coming in? Like, put on the ubiquitin, leave, and then a new E2 comes in, puts on the ubiquitin, and leaves?
Okay, so the question here is, if you're, and I'll just paraphrase it, tell me if I'm getting it right. Adding ubiquitins, is each ubiquitin being added by a new E2 protein? And basically another way of asking is, does E2 bind to multi-ubiquitins simultaneously? The answer is E2s all seem to just interact with one ubiquitin. So each time you're adding another ubiquitin, it's a new E2, which has been added the ubiquitin to it by E1, which then is going to give you that polyubiquitin chain. So it does seem to be multiple E2s that have to be coming in and out of that E3 complex. Okay, other questions on ubiquitin? Is there a question back there? Or just a stretch? Go for it.
After the E2 attaches itself, then it's no longer useful afterwards and it participates?
So the question is, what happens to the E2 once the ubiquitin has been transferred? So it depends on the E3 complex. Some of those, that E3 complex will sit there, wait for another E2 to come in and add more. Again, it's going to depend on your specificity. In some cases, if just a single ubiquitin is being added, then the whole complex will break apart. So it depends on the individual E3 complex for that. Okay, other questions on these ubiquitin processes? But again, basic idea here is that we've got these multiple enzymes. In order to understand how they're all working, you need to know whom they're talking to. And so this brings us to this ridiculously confusing and not understandable interaction map. But the basic message here is that all of these proteins are interacting with each other. And it's those interactions. Again, you're changing the interaction by putting a phosphate group on. You're changing the interaction by the G proteins modifying their structures. You're changing interactions due to the different E2s and the different E3s. So in order to understand how all of these things are working together inside the cell, you really need to know who all of the partners are. And this is just one example of a relatively simple case, remember correctly, this is yeast, of how each of these proteins are interacting with each other. And right at the end of the course, we'll talk about how you get this kind of map. But basically, any one of the dots here represents a single protein, and any line represents that it's attached to or interacting with another protein. So this dot right here interacts with that protein, with that protein, with this protein, with this protein, with that protein, with that protein. This is a relatively simple case. You got these proteins in the middle here that are interacting with a whole bunch of different proteins. And this is, again, not a surprising thing to look at, but basically to understand a lot of these interactions, you need a lot of math. And so one of the things I always tell people in this class is take more math classes, because if you really want to understand cell biology and molecular biology, you really need to understand these interactions. I can't understand these individual interactions by myself. I need to have a mathematical framework to lay on top of these things. So let's look at the example here of one of these ubiquitin modifying enzymes here. And this is just sort of zooming in to one of those parts of that interactome here. Down at the bottom, the cullen protein's still labeled in the same color scheme as we had when we looked at those structures a second ago here. So all of the lines here represent an interaction that this has with other different proteins. We talked about the F-box proteins. What are the F-box proteins doing? They're the interaction part of the E3. So they're your specificity. Any of these are going to interact with another protein, which is going to say this one is now going to get ubiquitinated through these kinds of interactions. And so just looking at interactions, and again, these are separate kinds of experiments that you can do, see how these proteins are interacting with each other. It gives you an idea on how the function is taking place. And in some of the cases in here, this is a particular example where people didn't know that some of these proteins were getting ubiquitinated, but they saw that they were interacting with each other, and just guilt by association, if ME130 here is acting with, MET30 and MET31 are interacting with each other, then you know MET30 interacts with the cullen. It means that MET31 is probably going to be being ubiquitinated. But just through these interactions, you can make predictions about what happens with a number of these different proteins. And it's just a way of thinking about how all of these things are interacting with each other, and through understanding how they're interacting gives you an idea of what the function is going to be. You'll have to check all of these interactions by doing separate experiments, but doing these interactome experiments, again, what interacts with what, actually relatively straightforward to do. It's relatively easy to make a picture that looks like this and allows you then to make predictions that you can test later. So that's the idea of these interactome studies. Okay, that's all I wanted to talk about in terms of proteins. At least for now. We'll talk about the individual proteins doing individual activities later on. I want to switch over to, well, not really my favorite molecule, but close to that. So, but before that, just to make sure our clickers are working properly, and hopefully we get closer to 100% here. The process of linking ubiquitin to a protein generally requires how many enzymes? 0, 1, 2, 3, or 4. Ten. 5. We're up to 152 clickers, so we're getting closer here. Okay, we seem to be, again, about evenly divided between 2 and 3. The key to this question is how many enzymes? What's an enzyme? What's it doing? It's catalyzing a reaction. What are the reactions we're talking about here? Ubiquitin, adding things to things. So what are the enzymes? E1, E2. Is E3 an enzyme? No, it's not an enzyme. So we just have 2. Turns out in some cases you actually only have 1 that's required, but that's a different story that we haven't talked about here. Okay, let's talk DNA. And I'll see if I can run things from down here. We'll see if it ends up working or not. So just a quick outline. We'll talk a little bit about the history of nucleic acid, why they're called nucleic acids, a little bit about the building blocks, adenine, thymine, cytosine, and guanine, Chargaff's rule, which is actually really kind of interesting because Edwin Chargaff never called it a rule. There's only other people who talked about it as a rule, and he actually didn't see that it was actually one-to-one correlations between the two. He said they were close to one-to-one, and then Watson and Crick said, you know, they have to be exactly 1. And Chargaff apparently visited them and said, oh, actually, I don't believe this. It's actually probably off. And so all kinds of interesting arguments that went on there. How DNA was shown to be the genetic material was actually shown very early on to be the genetic material in the 20s. But because there were some very strong personalities in the field at the time, people didn't believe a lot of the earlier experiments, and it was only really when people figured out that viruses were really the way to go. We had the Hershey Chase experiment, which definitively showed that DNA was the genetic material, at least for that one particular virus. We also now know that genetic material for some viruses is also RNA. And then we'll talk a little bit about the structure. That reminds me, the paper Watson and Crick, their first paper, the original model structure paper, is on D2L. I expect you to read it. There will be at least one question on the first midterm on that paper. So if you haven't read it already, read it. If you've read it already, read it again. So history of nucleic acid. Friedrich Michner, who was a Swiss biochemist in the late 1800s, was looking at salmon eggs in particular, because he was in Basel, which is on the Rhine, and at that point there actually were salmon that were swimming up the Rhine until all the chemical companies killed them, including one that I worked for. So he was just interested in what you saw in these salmon eggs. And what he saw was he could isolate this really big molecule that had a lot of phosphorus and was acidic. Now, why is DNA acidic? It's because of all of the phosphate groups on the outside of this model, which, by the way, has all kinds of problems with it. So come up and tell me what some of the problems are at some point. It's a really good review of thinking about DNA structure. But yes, the phosphates here, phosphates are losing hydrogens. And losing those hydrogens, that means it's acidic. So that's why you've got this acidic nature of this. People showed soon thereafter that chromosomes seem to be important for the genetic material. This is the rediscovery of Mendel's experiments that people ignored literally for decades. Saw that then chromosomes seem to be associated with genetic changes. People isolated chromosomes. They saw that there were lots of different proteins that were associated with chromosomes and a whole bunch of this, big thing that actually most people thought was a pretty boring scaffolding molecule. Just held all the proteins together, and the proteins were much more likely to be important for genetic information. And the reason for that really was, if you looked at DNA, it was actually really boring. It only had 4 different nucleotides that were associated. Proteins were exciting. They had like 20 different side chains. And clearly, since there's a lot more variability between you and me and all of the salmon, etc., the difference is in the proteins. It's not so much in the nucleic acids. And if you looked at the proteins in salmon look very different than our proteins. And Friedrich Michner, who's also working at the hospital, was able to get a lot of blood. He looked at that and said, hey, no, proteins are very different in blood than from salmons to humans, et cetera, as opposed to the DNA, which is really kind of dull and just about the same. So for a long time, and particularly Arnold Levine, who took nucleic acid and broke it into pieces, he said, C's, A's, T's, and G's are about all the same, kind of boring. How could that be genetic material if it's all the same? There's no information there, whereas proteins have a lot of information to them. So that was sort of the beginning part. And this is just an overview of this. Is DNA the genetic material? No way, it can't be. Only 4 nucleotides and probably just a repeating polymer, which is holding all the proteins together, and it's those proteins which are actually the genetic material. So how do people start to change or try to change some of this? And it turns out that these experiments were done. I don't expect you to remember dates here, but Levine said in 29 that it was just boring tetranucleotides, and Griffith did transformation. And basically this was, if there hadn't been this previous data, this would have been more than enough to prove that DNA was the genetic material. But since it was so boring, and Arnold Levine was this dean of biochemistry at the time, everyone ignored it. So what Griffith and coworkers showed was that transformation, and in this case, transformation, not alchemy changing lead to gold, was changing a infectious bacteria to, say, a non-infectious bacteria to an infectious bacteria. And so this is the basic idea. You had smooth bacteria, which, if you put them into a mouse, the mouse died. If you had rough bacteria, and you say, what do we mean by rough bacteria here? It's literally colonies of the bacteria, and we'll show you in just a second what those look like. Take rough bacteria, inject them into a mouse, the mouse is perfectly fine. What was really weird is if you killed off these smooth bacteria and put them into a mouse, the mouse is perfectly fine. But if you mixed dead with non-infectious, you still killed the mouse. So there's something in these dead bacteria which cause these live, non-virulent bacteria to kill the mouse. So this was really bizarre, and nobody really quite understood what was going on with it. But there's something in this so-called transforming principle. That then, later on, was picked up by Avery and co-workers, took this transforming principle and tried to figure out what it was, and basically just purified that transforming principle. And I promised I'd show you what smooth and rough look like. These are the smooth colonies of bacteria. These are the rough colonies of bacteria. So if you take dead ones of these and mix them together with these actually not only become pathogenic and kill the mice, they also start to look like this. So it's nice you didn't have to kill all the mice in the process. All that you needed to look at, and this is what Avery's lab did, was to look at bacteria that were like this and trying to get them to change into bacteria that looked like this. We now know that it's a capsule gene. The capsule is basically making a slime that's on the outside of the bacteria that makes them look like this. And the process of heating up the cells released the DNA, which then changed the DNA in these guys, made them to be slimy. There was this slimy aspect of the cells that actually allowed them to get into the mouse and infect them and cause the mice to die. So how did Avery show that this transforming principle was actually DNA? Basically, he took advantages of the enzymes that people knew about. Remember, ACEs, right? These are enzymes. And again, for the most part, was an ACE do. It caused hydrolysis of whatever that ACE is hung on the end of. So DNase breaks down DNA. RNase breaks down RNA. Protease breaks down proteins. What they found was that if you treated with RNase, the transforming principle was perfectly happy. You treat it with protease, the transforming principle is perfectly happy. What happens if you treat it with DNase? No more transforming principle. So that's all gone. So this was the evidence, again, in the 40s that DNA was the genetic material. And again, nobody believed them. And there's a great story about Avery giving this presentation right after somebody was talking about Dramamine. And the whole audience clears out before he talks about this transforming principle because nobody cared. So kind of the right experiment at the wrong time. So one thing that happened a couple of years later was Chargaff, and Chargaff was just looking at the different amounts of adenine, thymine, cytosine, and guanines in different nucleic acids. And he had basically developed techniques where he could be much more precise about the relative amounts of all of these different nucleotides. And what he found, basically through the study of bacteria, that there were some organisms where A, T, C, and G weren't actually identical. But A's and T's had almost the same concentration, and C's and G's had almost the same concentrations. And if you go back again and look at his papers, you'll see that it's not quite one, but just very close. And the real key to this is that in some cases, A's and T's were not the same as G's and C's, and this all came from the studies of different bacteria. If you look at salmon and humans and plants, the numbers are almost identical in terms of A's, T's, C's, and G's. They don't vary at all. But if you look at bacteria, you can get very different amounts of G's and C's. In this particular case, I think it was the tuberculosis bacterium that they were looking at. High GC bacteria. So this really kind of laid the groundwork for Watson and Crick and our model here. But there was still a lot of controversy about DNA was really the genetic material. Now, if you remember the timing here, this was late 1940s, moving into the 1950s. What happened around that time historically? Atomic bomb, exactly. So a lot of study of radioactivity and understanding and actually being able to get various different radionuclides, which actually turned out to be really easy to detect and differentiate from one another. And so this also was about the time that people decided to study viruses, and particularly viruses of bacteria. And if you're interested in more about this, take my virology course next term. We'll talk about this probably ad nauseum as far as you're concerned there. But the neat thing about viruses, and this is the physicists who jumped onto this, a lot of the physicists who had been working on the Manhattan Project or various parts of it, who were then out of a job because that project was done, they wanted to find something else to do and said, okay, we figured out everything we need to know about nuclear reactions, let's figure out biology. But since they're physicists, they said, let's study a simple system to try and understand biology. And so they found these viruses, which were very simple systems, and probably most importantly here is that these viruses were just made of protein and nucleic acid. There wasn't any of those annoying lipids and sugars and so on and so forth, which are there. So protein and nucleic acid only, but these were clearly heritable. So if you had one kind of virus that stayed that same kind of virus, you could also have mutations in that particular virus, see those mutations in terms of their infectivity. And it was shown that they could only replicate inside of other cells. They were obligate intracellular parasites. And that was known also because of the development of the electron microscope, which happened almost at exactly the same time as all these developments with the various different radionuclides, etc. So they picked this one particular bacteriophage, or bacterial virus, bacteriophage T2. Most people talk about T4. Turns out T2 and T4 are extremely similar to each other, known as the T-even bacteriophage. When you have an infection process, the phage structure stays on the outside of the cell. The DNA, or whatever the genetic material is, goes on the inside of the cell. They replicate by bursting these cells open. And these are some beautiful color EMs, which have just been colorized afterwards. Here are a few of these bacterial viruses. These guys still have their DNA in them. This one has lost its DNA inside the cell. So I think there's also a nice example here of where the people who did the work actually got the credit. So most people talk about the Chase-Hershey experiment. No, Hershey-Chase experiment, but it really should be Chase-Hershey because she did most of the work. And there's a great story about Martha Chase, who apparently she'd done the experiments, they gotten the results. Al Hershey had talked about them, and she'd gone out to a dance afterwards and was talking to one of the other people at Cold Spring Harbor and said, basically, I just keep doing this work and I'm an underpaid technician, why should, none of this stuff is really exciting. And then the other people there said, actually, this is kind of exciting. You're probably going to be famous, which she now is. So Martha Chase did almost all the work. Al Hershey was just a two-person lab, and she was the technician who was working in the lab, also get the credit for the paper as well. So what did Martha Chase do that Al Hershey told her to do? Basically took bacteriophage and bacteria, mix them with the radionucleotides, which are available because of the Manhattan Project, and mix them with S35 or P32. Why S35? Sulfur. Do you see any sulfur in here? No. Do you see phosphate in proteins? Yes, but very small amounts of phosphate. S35, you're going to label just proteins. P32, mostly nucleic acid. Infect the cells. Separate the virus particles from the cells is the blender experiment, the E. coli daiquiri. And then just look, where's your radioactivity? P32 and S35. And as they showed very nicely, the P32 is mostly in the cells and the viruses of the following generations, and the S35 remained in the virus particles sitting on the outside of the cell and didn't get passed along from generation to generation. So just a pictorial version of this. Here, there are actually two different experiments. We're not going to get to this clicker question, sorry. We'll start out with the clicker question next time. Here, the two different experiments where you had a bacteriophage, bacterial virus labeled either with P32 or with S35, two separate experiments here, where you have the S35 does this infection, the phage is removed again through this blending experiment. You look at what's left in the cells after you've removed these virus particles. You see that there's no S35 associated with the cells. It's all associated with the virus particles. If you look at P32, you notice that it gets transferred. And so the P32 is because we've got all of our phosphorus in our DNA. The phosphorus in the DNA, again, gets transferred. That's your genetic material. The S35 does not. So we'll stop here, the clicker question, and then we'll talk about Watson and Crick on Friday, so you have an opportunity to read the paper. Again, shouldn't take you more than about 5 minutes.